All right, welcome to another webinar of the FARA Flash Talks. I'm Liz Sorani, Director of Research at FARA. We hope you've been participating in our webinars and celebrating with us at the month of May, the FA Awareness Month. Each week on Thursdays at noon, we are bringing you some great examples of FARA-funded research presented by junior investigators from all over the world. The presenters have 5 minutes to explain to us in lay terms their area of research and their project. After each presentation, you can ask questions by typing them in the Q&A box, and please use the Q&A box and not the chat box to ask your questions. We are recording the talks and we'll be posting them on YouTube, and the previous Flash Talks are already on YouTube. At the end of the webinar, we'll ask you to complete a short survey, only 3 questions, where you can rate the talks based on how effectively the science was presented, how understandable it was, and how each presenter was able to communicate the impact of their work on our understanding of the disease and the development of treatments. And the highest presentations, the highest rated presentations will receive an award. Our moderator today is Caroline Spencer. She'll be introducing the speakers and take your questions. Caroline, please introduce yourself and take it from here.
All right. Thank you, everyone, for joining us today. I'm Caroline Spencer. I'm an FA patient here in the U.S. I live in Massachusetts, and I am a speech researcher. So I think we'll go ahead and get started. So our first presenter today is Valentine Musbach from the Institute Neuro-muscular, and she'll be presenting a presentation on partial frataxin bypass on iron sulfur clusters, biogenesis, in murine cellular model.
Okay, thank you. So I'm sharing my screen. Okay, is it okay?
Mm-hmm.
Okay, so hello, I'm Valentine. I'm a postdoc in Helen Puccio team. And today I will talk about my project on frataxin bypass in murine cellular model. So to understand this project, first you need to know what is an iron sulfur cluster. So an iron sulfur cluster is an assembly of iron and sulfur that is required as cofactor for many proteins that are involved in key cellular process in the cells. For example, there is plenty of proteins involved in mitochondrial function that need an iron sulfur cluster to work. So how the cell produce them and distribute them to their target is thanks to a specific machinery that acts as a factory and that is localized in the mitochondria. So this factory is composed of different proteins. Each proteins have a specific role. So the ISQ protein is a protein where the iron and the sulfur is assembled to form the cluster. The NFS1 protein will provide the sulfur. The iron donor is still unknown, and the frataxin protein will play a regulatory role by accelerating the production of iron sulfur cluster. So any kind of deficiency in this factory can potentially lead to important cell dysfunction. And it's well known, for example, that in Friedreich ataxia, a low level of frataxin protein leads to a decrease of production of iron sulfur cluster. This mainly leading to a mitochondrial defect in the cells. So this is what happens when we have a low amount of frataxin. But now what is happening if we completely remove frataxin from the cell? So if you're doing that, for example, in a yeast, you will end up with severe growth defect of that organism. But even worse, when you completely remove frataxin in a mammalian system, so for example, in cell culture, cell in Petri dish, or in mice, it's lethal. But there is a recent study that identifies a specific mutation in the ISQ gene that lead to the expression of a specific variant of ISQ. sorry, I lost my mouse. Yes, it's here. So a specific variant of ISQ. So this variant is different from the wild type just by a difference of one amino acid. And the expression of this variant in a yeast can turn the yeast into a frataxin-independent organism. That means that even in absence of frataxin, the yeast have a normal growth. So the question is if this variant of ISQ can bypass the lethality due to the lack of frataxin in a mammalian system. So to answer to that question, I first use a cellular model. So it's murine cells in culture that I engineer to introduce the specific mutation on the ISQ gene to be able to have cells that express the variant of ISQ. Then I remove completely frataxin from these cells, and I observed if I was able to obtain cells that was alive. And it is the case, as you can see here. However, these cells, if we compare them to the wild type, have a very slow growth curve. Furthermore, where I tried to investigate the phenotype of these cells, I could still notice that they present a strong defect in the mitochondria. As you can see here in the microscopy picture, so in wild type cells, you can observe healthy mitochondria. Same for the cells that express the variant of ISQ and still have frataxin. But the cells that only express the variant of ISQ, you can notice that they have a lot of empty mitochondria, like dead mitochondria. So finally, for now, the conclusion is that this variant of ISQ can indeed bypass the lethality of the absence of frataxin, at least in culture cells. But our data suggests that the iron sulfur cluster biogenesis is still strongly impaired because we have mitochondrial dysfunction. But all that made a very good cellular model to try to better understand how the iron sulfur cluster biogenesis is working and how it is regulated in the cell. Moreover, as an ongoing work, I'm currently look at the effect of this variant but of ISQ, but this time on neuron in culture, because it's the main cell type affected in FA. And I am also trying to test if this variant of ISQ is able to bypass the lethality of the absence of frataxin in a mice. So for that, I have a mice that is expressing our variant of ISQ that I will cross them with different FA model mice. And all that, again, will make good models to study at the fundamental level the pathway related to iron sulfur cluster biogenesis. Thank you.
Okay, thank you very much. Let's see if there are questions. There's one. Can you repeat the purported MOA mode of MOA of the M141 mutant and the implications on the activity of the FXN protein?
Okay, so
did you understand the question?
So yeah, I don't understand the part with the MOA, but for the implication on the activity of frataxin protein, so we are on a system that doesn't have frataxin anymore. So I don't know the impact of the mutation on the activity of frataxin. But what I can tell you is that I'm currently working with a team in Grenoble in France that is checking the activity of all the factory of the complex in absence of frataxin and with the variant of ISQ.
Another.
So I did a preliminary experiment. So it wasn't with FA human cells, but I tried to introduce this mutation in iPS cells, but that are from control person, so not not from a FA patient, but I just managed to introduce the mutation in the iPS cells. So I didn't delete frataxin yet in these cells, so I don't have the answer to your question, Jennifer.
Yeah, so the question was, have you looked
Oh,
at...
yes, sorry. I was looking at the chat. I forgot to say the question. So the question was, have you looked to see if the ISQ mutation rescue or improve FA human cells? So I didn't put that mutation in FA human cells once again, but I tried to put it in an iPS model from control. And I managed to introduce the mutation, but I didn't delete frataxin, so I don't have the answer for that. Yeah. For now.
So sorry if I missed this, but what part of ICU is the mutation? Is it the one that interacts with NFS1, or
So as I say, the difference with the wild type is just one amino acid, and the position of that amino acid, what is very interesting and a bit strange also, is that it's not at the site of interaction with NFS1. It's a bit aside. So it's a bit like, again, a mystery for now, how this single mutation can still manage to restore a viability of cells in terms of interaction with the complex. But we hope that with the biochemical data, we will have more insight on that.
Recording in progress.
Oh.
All right.
Oh.
I think we will have to move on to the next.
Hold on 1 second.
We have an echo. There we go.
I think Carl might be on
yeah.
more than
I just
once.
muted him. Yeah.
Okay.
Go ahead, Caroline. Sorry.
So our next presenter will be Yina Dong from Children's Hospital of Philadelphia. I'm presenting on frataxin controls ketone body metabolism through regulation of OXCT1.
Can you stop sharing, Valentine?
Oh yeah, sorry. Where is the? Sorry. Floor is yours.
All right. Oops. Okay.
Okay. Good afternoon, everyone. And first, I want to thank FARA for giving me this opportunity to present our work. So today I'm going to talk about the ketone body metabolism in FRDA. So ketone body are alternative energy source at low glucose levels, such as starvation or prolonged exercise. Ketone body, which contains acetone, beta-hydroxybutyrate, and acetoacetate, is mostly made in the liver from fatty acid through a series of chemical reactions. Once it's made, ketone body enters the bloodstream and transported to metabolic active tissues, such as the brain and the skeletal muscle for utilization. In tissues, ketone body utilization is controlled by OXCT1, also called SCOT, which is a rate-limiting enzyme, converting ketone body to acetyl-acetyl-CoA, which is then converted to acetyl-CoA, entering TCA cycle for ATP production. Excuse me. So why do we study ketone body and what happens to ketone body metabolism? So using proteomic approach and biochemical studies, we found the ketone body, we found the frataxin interacts with OXCT1. This is mass spec results showing that frataxin can pull down OXT1 from mouse cortical homogenates. Frataxin also regulates the level of OXT1. This is experiment we done in Kiko mice, which is frataxin knocking and knockout mice. Frataxin levels are dramatically decreased in this mice. And we also found decreased OXT1 levels at both cerebellum and skeletal muscle at symptomatic age. Similar decrease also occurs in FRDA patients' skeletal muscle. So does this decrease alter ketone body metabolism in FRDA? So to examine this, we fasted control mice and Kiko mice for 24 hours, and then measured the beta hydroxybutyrate in the blood. As you can see, like 24 hours fasting can lead to significant increase of beta hydroxybutyrate in both control and Kiko mice, but the level of beta hydroxybutyrate is significantly higher in Kiko mice compared with control mice. This indicates that OXT1 deficiency could trigger ketone body utilization deficit, leads to increased beta hydroxybutyrate in the blood. To confirm this, we knocked down frataxin in C2C12 cells, a skeletal muscle cell line, which leads to decreased OXT1 levels. As you can see, this is bar graph, this is Western blot. So when we treated these cells with beta hydroxybutyrate could not be metabolized into acetyl-CoA as what happened in control cells. This occurs in both no glucose and glucose-containing medium, which is equivalent to fasting and non-fasting condition in animal model. Accordingly, succinyl-CoA, the level was significantly higher in frataxin deficient cells. That suggests decreased consumption by ketone body. So this results indicates that frataxin deficiency caused OXT1 reduction can trigger ketone body utilization deficit. To summarize, our present study demonstrates a new role of frataxin in ketone body metabolism. This role is mediated through the regulation of OXT1. So we also found a compromised ketone body metabolism, which may explain the clinical feature of FRDA patients, such as exercise intolerance and prolonged post-exercise recovery. Our current study also doesn't support the use of ketogenic diet for FRDA. Thank you.
Thank you very much. All right. We have some questions.
I can start with one. So is OSCT1, OXCT1, is it more expressed in certain tissues? Do you know?
Yeah, it except liver, because liver make a ketone body, so liver doesn't have OXT1, but OXT1 expressed in a lot of other tissues. Yeah.
Is it more expressed in tissues like, you know, the heart or the skeletal muscle?
Yeah, metabolic active tissues, like you have more OXT1 expression.
Yeah. We have two questions. One, does OXT1 contain an iron sulfur cluster?
Not as I know.
And is OXT1 decreased in expression or lost through turnover?
So we did experiment. We found like OXT1 degradation was increased without frataxin. So frataxin can protect OXT1 from degradation.
Thank you. We have another question. If a ketogenic diet is ruled out, what might be other routes of benefit for this understanding of effects and role in regard to ketone metabolism?
So, sorry, can you repeat your question?
If the ketogenic diet is rolled out, what might be other routes of benefit for understanding the effects and role in regard to ketone metabolism?
So is there another diet that would be better?
So, well, I would, because like ketone body metabolism is compromised because of the lowered OXT1 levels. So I would suggest more like a fat like intake. I would suggest more like carbohydrates. Yeah, because like when you have more fat, you have more ketone, you get like you can cause ketoacidosis, like leading to severe symptom for patients, which if they cannot like metabolize ketone bodies. Yeah, I would suggest like more like carbohydrates, like not too much fat. Yeah.
All right, thank you. Excuse me. We will go to our next presenter. Second. There we go. All right, our next presenter is Camila Weitzel from the Children's Hospital of Philadelphia, and she'll be presenting on now recruiting the FA Sweetheart study, glycemia and echo findings in Friedreich's ataxia.
Let me share my screen. Okay, so good afternoon. I'm Camila Weitzel, and I'm an undergraduate student studying bioengineering at the University of Pennsylvania. And I've recently started working with Dr. McCormick and Dr. Tamroff at CHOP, and I'm excited to share with you what I'm working on. So today, I will be talking about the relationship between cardiac characteristics and glucose or sugar levels in both children and adults with FA, with and without diabetes. So studies have shown that there is a relationship in people without FA between diabetes or even pre-diabetes and heart disease, including cardiomyopathy. We know that folks with FA are at risk for diabetes or abnormal glucose levels and are also at risk for cardiomyopathy. As far as we know, no one has looked into the relationship between FA-related diabetes and cardiomyopathy. With a better understanding of the potential relationships that could exist, we can maybe change the way we screen and even manage diabetes or pre-diabetes and FA in the future. So this is an actively recruiting remote study to initially evaluate this relationship, with additional ongoing studies assessing more detailed evaluations of the cardiac and glucose parameters. In this study, participants wear continuous glucose monitors, which are used in clinical practice to monitor a patient's glucose levels. The participants wear the CGMs for up to 2 weeks so that their glucose measurements can be taken remotely. As you can see, this is Dr. Tamroff wearing a glucose monitor on her upper arm. Down below, this green picture shows the glucose monitor before it is placed. And here at the top, this shows the small monitor, which is slightly bigger than a quarter, and the monitor that is used to start the monitoring. The glucose monitors are placed at home over a video chat with Dr. Tamroff. This study requires participants to be diagnosed with FA, enrolled in FACOMS at CHOP, and have had a recent clinical echocardiogram. Both participants with and without diabetes are welcome. If you look next to the green picture, you will see a picture of a glucose tracing. In this image, each color represents a different day. This tracing here shows that the participant mostly stayed within the target gray zone, a range between 70 and 140. In the hours where the participant was sleeping, their level remained relatively stable. When the participant was eating, their levels fluctuated a bit, but still mostly remained in that gray zone. So far, we have had 30 participants wear the glucose monitors as part of this study or Dr. McCormick's ongoing study evaluating exercise in FA. And if anyone would be interested in hearing more about that study, make sure you listen to my colleague Nikki Salenti's talk from last week. So about half of the participants were female, with ages ranging from teenage to young adult, and with relatively normal BMIs. The average within participant glucose level was 85, with a coefficient of variation, a commonly used marker of glucose variability, equaling 19%. These are pretty much what we expect from normal glucose tolerance. So up to this point, most people in the study do not have diabetes. So when excluding the individuals with diabetes, these numbers were similar. So we are interested in enrolling more participants with diabetes to more closely compare these numbers. We are not seeing a high rate of high glucoses in individuals without diabetes, with a median percentage of time spent over 140 being less than 1%. The rate of low glucose levels, or levels less than 54, has also occurred infrequently. That was the glucose level part, but now the next part of this study looks at the cardiac portion by reviewing echocardiograms. Here's another picture on the slide demonstrating someone receiving an echocardiogram. So in a subset of individuals who had this CGM data, we have clinical echo data already available. With the echoes, we looked at multiple parameters assessing heart size and function. Two of the measures of heart size are septal wall thickness and posterior wall thickness. When assessing these parameters, we can look at the z-score, which is normalized based on age, height, and gender. And when the z-score is greater than 2, this is indicative of hypertrophy or increased cardiac size. Half of individuals had a septal wall z-score and or a posterior wall thickness z-score that was greater than 2, indicating some amount of hypertrophy. We have not yet fully evaluated if there is a relationship between these measures of glycemia and measures of heart size, since we have not evaluated enough participants or enough echo results yet, but that is our next step. So far, we have concluded that remotely replacing the CGM is feasible, and we have not found a high rate of high glucose levels, as people without diabetes do not have high levels of hyperglycemia. So far, the data is what we've expected based off of other studies that looked at various populations also at risk for diabetes, like individuals with cystic fibrosis. As expected, we have found about half of individuals with some amount of cardiac hypertrophy. So if you're interested in this study, please reach out to Dr. Tamroff. And I'd like to thank you all for listening today. And I'd like to thank FARA for this experience, everyone at CHOP for their help in this study, and a big thank you to all of our participants. Thank you.
Thank you very much, Camila. Let's go to our chat. We can take questions for Camila about the study.
So maybe I'll ask one. So these patients, they have so they're considered diabetic when you measure their A1C. Is that how you know, you measure the A1C level, and you know, if it's above a certain, it's like, you know, regular diabetes when you check their A1C level. And I was wondering if, you know, they are all the one that are, you know, maybe pre-diabetic, they all progress to become diabetic or not, if you know that.
Are you asking if, could you repeat the last portion of your question?
Yeah, so if, you know, I assume that it's the same, you know, that you measure the A1C level to you know, check their diabetes. Is that correct, or am I wrong? And
I'm pretty new to this study. If with any like really specific questions, you have to ask Dr. Tamroff, but yeah, just probably better to ask Dr. Tamroff.
sure.
Camila, I have a question. What is the enrollment goal and how many people do you still need?
I believe our enrollment goal is 40. So we need, so far we have evaluated 30 participants, so we need more, 10 more.
I think there's another one. Do you see the other one?
Mhm.
Yeah.
Are continuous glucose monitors routinely used in FA patients with confirmed diabetes? Or do they usually test blood glucose via finger prick test?
I'm not sure what like the most common thing is, but I think it's probably a mixture of some people use the monitors and some people do a finger prick.
Any other questions? I think we're good. Thank you, Camilla.
Thank you.
All right. So our next presenter is Annika Varma from the University of Rochester, and she will be presenting on development and validation of the patient-reported and caregiver-reported FA health indices.
Hello, everyone. My name is Annika Varma, and I'm a clinical research coordinator with Dr. Chad Heatwell's Clinical Outcomes Group at the Center for Health and Technology at the University of Rochester. It's a pleasure to speak to you all today about my group's work on the development and validation of the patient-reported and caregiver-reported FA health indices. So this work was generously funded by FARA, and I'd like to thank them for their support. So my team's work centers on developing disease-specific patient-reported outcome measures, or PROs, and caregiver-reported outcome measures, or CROs, for a variety of disease populations. We have developed more than 100 such measures, which have been used by academic partners, industry, government agencies, and foundations in patient monitoring and clinical trials worldwide. So stepping back a moment, what exactly are PROs and CROs? A PRO or CRO is any report of the status of a patient's health condition that comes directly from the patient or caregiver. These measures provide a voice for patients and caregivers to provide direct insight into FA health and into potential therapeutic benefit. Furthermore, PROs and CROs measure concepts that are not measurable in other ways. They are accepted by the FDA for use in drug labeling claims. They evaluate health in the areas most important to patients. They can be completed by a patient or caregiver without external administration, and they track small but clinically relevant changes in health. Essentially, they transform patients from the center of focus into active participants in evaluating the efficacy of therapeutics. So my team and I recently completed development and validation of the FA health indices, which include the FA-High and the FACR-High. The FA-High is a PRO for patients with FA ages 8 and up to complete on their own. It includes 113 individual symptoms that cover 18 symptomatic themes or subscales. The FACR-High is a CRO for caregivers to report on pediatric patients with FA ages 5 and up. It includes 84 individual symptoms that cover 18 symptomatic themes or subscales. So in developing the FA-High and FACR-High, we started by conducting initial qualitative interviews with individuals with FA and with caregivers. We asked open-ended questions about which symptoms of FA are most important to patients. We analyzed the interview responses and then constructed 2 surveys encompassing all the recurring important symptoms, one survey for patients and another for caregivers. Next, we conducted 2 cross-sectional studies in which individuals with FA and caregivers completed our surveys to rate the impact of each symptom on daily life. So the graphs shown here show the prevalence and average impact on a scale of 0 to 4, with 4 being the most severe of the symptomatic themes asked about on the patient and caregiver surveys. So as you can see, impaired coordination, inability to do activities, limitations with mobility and walking, and fatigue were the most prevalent symptomatic themes. So using the data from the cross-sectional study, we developed the first versions of the FA-High and the FACR-High. We selected questions from the cross-sectional study that were considered highly relevant to those with FA, of clear and consistent understanding, applicable to a broad population of patients, and amenable to therapeutic intervention. We then conducted beta interviews with individuals with FA and with caregivers to assess the comprehension and usability of the PRO and CRO. And then as you can see by the quotes here, interviewed participants generally found the FA health indices to be well structured and relevant to them. After beta interviews, we performed test-retest reliability evaluation, where participants completed the FA-High or FACR-High once at baseline and then again 2 weeks later. And results showed that the 2 health indices are highly reliable. After test-retest reliability, we formulated the final versions of the FA-High and FACR-High, and determine the internal consistency and scoring algorithms. We also conducted known groups analysis where we compared the scores from the FA high and the FACR high between groups of patients with FA believed to differ in disease severity. So that pretty much sums up our development and validation processes. The FA high and FACR high are now complete and ready to be used in clinical monitoring and therapeutic trials. Additionally, our team was recently awarded continued funding from FARA to perform longitudinal evaluation of these outcome measures. So with that, I'll conclude my talk. Thank you all for listening and supporting the FA research community. You make our work possible and allow us to bring your voice to the forefront of research and clinical studies. So I hope you will continue to join us as we go forward with our new study, and I'd be happy to answer any questions.
Thank you very much. Set our Q&A box.
Annika, do you want to tell us a little bit of what you're going to do next in the next phase, the longitudinal study?
Sure. So in our longitudinal study, we are going to implement the FA high and the FACR high, and we're going to compare it with a few generic outcome measures like the SF36 and the PEDS-QL to determine, patient preferences and the usability of this instrument. We'll also determine the MCID, which is the minimal clinically important difference, which will be helpful when we implement this in clinical trials to show if there's a significant difference when patients are on a therapy or to show disease progression. So if there's a meaningful difference over time. And in the longitudinal study, we'll have patients and caregivers complete these patient-reported and caregiver-reported outcome measures, at baseline, 6 months, 12 months, 18 months. So it'll really track disease progression or therapeutic gain over time.
And just a curiosity, how long does it take to answer all these questions? There's a lot of questions.
Yeah, so there are 113 questions on the patient-reported outcome measure and 84 questions on the caregiver-reported outcome measure, but they're really designed, it's like a list of symptoms and then kind of a rating scale. So it's really a minimal patient burden. It usually takes 15 to 20 minutes to answer all the questions. So it's not as burdensome as it may seem at first.
I have a question. Have you looked for differences in symptomatic impact in different subgroups, like pediatric patients, stage of FA?
Yeah, so we have done that. We've done known groups analysis where we compared the scores from the FA high and the FACR high in different subgroups of patients. So like based on age, sex, functional staging of ataxia, ambulation status, all that kind of thing. I didn't have room on this slide to show it here, but we did do a comparison in different subgroups.
Any other questions? I think that's it. Thank you, Annika.
All right, thank you so much.
Let me get back to my other screen. All right, so our last presenter today will be Elliot Siwell from the University of Buenos Aires. He will be presenting on finding structure function relationship in the iron sulfur cluster assembly machinery. Carl, are you there? All right, we're able to.
Okay, I'm here. I'm sorry. Is my screen sharing?
Nope.
Not yet.
Let's see. Now?
No, we can't see it.
God. ¿Se está transmitiendo algo? Lo que sea. O sea, ¿qué ves, Core?
No sé.
Carl, do you want to email me the presentation and I can share it? Oh, perfect.
Okay.
No.
Seems to be working now.
Yes.
Okay, is this light on? Because I don't see it in my screen.
Oh,
Yeah,
your
your share.
presentation was there and no, it's not.
So you're sharing the Zoom. I don't know how is that possible, but you're. There it is.
There it is.
Now?
Yes.
There it is. Okay, I'm so sorry.
Yay.
Yeah, I have a few troubles today. I'm so sorry. But now we're here. So my name is Elliot Siwell. I am a PhD student at Javier Santos Lab at the Universidad de Buenos Aires, Argentina. I have been working on proteins related with Friedreich ataxia since 2018, mainly trying to understand the molecular role of frataxin in the context of the iron-sulfur cluster supercomplex. As a reminder, frataxin exercises its function in relationship and cooperation with at least another 5 proteins. Well, that together build a bigger and functional structure that is what we call a supercomplex. Of course, and for context's sake, it is worth saying that ISCU, frataxin, and NFS1 are some of these proteins that belong to the supercomplex. So today, what I would like to share with you, it's a computational method we have implemented for movement predictions in proteins and a summary of our findings. So we used molecular dynamic simulations applied to the entire supercomplex in the presence and absence of frataxin to try to better understand the role of frataxin might have in its function and its movements. This technique is about starting a starting structure. For instance, let's say the puppy picture we have left, which is static because it's a photo of a puppy, and so we build that photo into a computer, and we build that photo into a computer and start assigning it some movement and forces based on the laws of physics. So by doing so, So what we do is to apply the laws of physics to the puppy picture so we can predict the movement and forces that apply to him and what movement should it have according to those laws. So by integrating that, we can build up a new picture of a new puppy in a new position. That's what we have on right side, and do it that again to predict a new movement and a new forces and a new picture of a new puppy. So by doing that enough times, we start building up a collection of frames and we can compile enough to have a movie. So at the end of the process, we have a movie of a very likely moving puppy that is deduced somehow from the starting structure, which is our puppy at the left. So back to our supercomplex, we apply these principles to the structure. And particularly, we want to see what happens in the places where frataxin, ISCU, and NFS1 interact with each other. This is what we have at our bottom left signal ISCU and frataxin, and in blue, we have NFS1. And so we can see there is a place where the 3 of them interact strongly with each other, especially the last part of NFS1, what we call the C-terminal section. And what we found as a general feature is that the C-terminal section of NFS1 has a very variated kind of movements. It moves more when comparison with the presence of frataxin, which means frataxin helps anchor that portion of the protein, and it is more static, for to say it somehow. Interestingly, what we find is that when we mutate the protein at NFS1 at this portion, at the C-terminal portion, we find that there is a lot of loss of function in those in this section when we have a punctual mutations of these amino acids. And so the closing conclusion would be that frataxin modulates the dynamics of the members of the iron sulfur cluster supercomplex. It sets ISCU in a suitable position for reaction and anchors the C-terminal portion of NFS1 that enables the assembly of the catalytic site. And that mutation of this stretch of NFS1 results in a significant decrease in the activity. That'll be more or less all.
Thank you very much. All right, now we're gonna open the floor to questions.
So Carl, are those mutations in the flexible loop that moves in NFS1?
No, you mean the one that has the catalytic cysteine?
Right. Yeah.
No, this portion would be the C-terminal portion, which means, for instance, histidine 457, which is the last amino acid of the chain, and let's say the 10 before it, or maybe less even.
But these mutations prevent this movement, the movement of the loop. Is that correct? Or I didn't.
No. What we saw at the dynamic at the movie is that when frataxin is present, this portion of NFS1 is more static. It moves less.
Oh, okay. So the C-terminal moves less.
Yeah, the C-terminal moves
okay.
less.
Okay.
And when we mutate the C-terminal, we have less activity too.
So do you think this can be targeted somehow and to improve the activity of the complex?
Well, what we think this far is that this portion is important for the reaction, and that maybe somehow it's part of frataxin's function to fixate it there. Maybe for special or specific interactions with ISCU, or just to allow better movement of the catalytic loop. But we honestly don't know if it would be targetable yet.
Thank you very much. We are happy to take, there's one question here. Thinking back to Valentine's talk, would you predict that the SQ variant she was working with might alter the association of SQ with the NFS1 in the absence of frataxin into a more favorable arrangement that resembles the arrangement of when frataxin is present?
I'm not sure if I understood correctly. It would be like, do I think that ISCU variants presented as those presented for by Valentine would?
Oh, yeah. So I think the question is, thinking back to Valentine's talk, if the SQ variant that she was working with,
yeah, if that makes, you know, the complex into a more favorable arrangement, so that, you know, that can doesn't necessarily need frataxin. So if that mutation
okay, I get it now. That'd be actually a very interesting question, but the only way to figure it out is actually building up that complex and simulate that, or characterize it. You know, we could use other biophysical techniques such as CD or like those.
Yep.
But interesting question though. I might talk to Valentine's later.
right, I have a technical question. What is the name of the force field for your MD stimulation, and how long was it? And what size of the cell do you use for your simulation?
What size of what, sorry?
The size of the cell for your MD simulation.
Well, the force field was amber 16. And the size of the cell, I can't remember the size number, the exact number right now, but it was a very big one. Because it needs a very big one. It's a very big structure. It parted it from that. It was big.
Any other questions? We have just a few minutes left.
Okay, I don't think that we have any more questions. All right, so I would like to thank all the speakers today once again for their presentations and also their contributions to the FA research. Thank you, Caroline, for moderating. Remember to keep your browser open to vote at the end, and please join us again for the last session next Thursday, May 26th at noon Eastern time. Thank you.